Randomised controlled trials are the best and only reliable way to find out if a new treatment or prevention approach works. But the usual approach to randomised controlled trials has some important disadvantages.
Traditionally, how we've done the testing of new treatments is to assess the new treatment and compare it against a standard or a control treatment. And that's led to what's called a two-arm trial, comparing the new versus the control or standard. The challenges with that is often it takes a long while to get to the result, and often they show that the new treatment is no better than the control. We're in a situation now where we have many new treatments that we might want to evaluate. So we need to think differently than just comparing one new treatment against the control, especially if there's a reasonable chance that that new treatment might not be better than that standard.
If it turns out that you've got a number of different drugs that look interesting in phase 2 and you set up a number of different phase 3 trials, these trials will be competing with each other. You'll have potentially the same control arm running in a number of different trials, and so that the trials are slowing each other down, and time is money in this context.
To tackle these problems, researchers have developed an innovative new trial design, the multi-arm, multi-stage trial, a MAMS trial for short.
So the first bit of that is multi-arms. So instead of having just two arms as a new treatment versus a standard treatment, we just have many arms, many new treatments against a standard treatment. And you can quite quickly see that if you're testing many new treatments against a standard, the possibility that one of them is going to be better than the standard increases. It's like doing many two-arm trials which are often done back to back, all at the same time.
And in the multi-stage approach, we stop recruitment early to those agents which are looking insufficiently interesting based on pre-planned criteria. So that programmatic approach, this programmatic MAMS trial, is better than the traditional approach because it requires fewer patients and less overall time. And that's because we've got concurrent assessment of a number of agents. Because we're randomizing from the outset, we can make comparisons from the very outset. And actually we have one seamless trial and an awful lot less bureaucracy, a lot less of the administration by putting this all into one protocol.
A general rule of thumb that we're developing in this area is you buy 3 and get 1 free. So basically if you make 4 comparisons, 4 new research arms against a standard arm, then one of those is effectively free.
One example of a trial using the multi-arm, multi-stage design is the STAMPEDE prostate cancer trial.
STAMPEDE is an example of a multi-arm, multi-stage trial, and this graphic shows figuratively the design of STAMPEDE when it started. From 2006 up to 2022, volunteers could start into the trial in October 2005, at which time we had 1 standard of care arm and 5 research arms. The 5 research arms each tested an individual drug in arms B through to D there, or combinations of those drugs, arms E and F.
A key element of the multi-stage approach is having a number of pre-planned reviews of the data, or interim analyses, as the trial progresses.
So we set the trial up with interim analyses because the primary endpoint we wanted for STAMPEDE was overall survival. That obviously takes a long time. We don't want to carry on recruiting and following up large numbers of patients if it's futile. And all of the initial 5 arms got through the first interim analysis. And it's worth thinking of it like a high jump contest. The bar keeps going up, so they got through the first hurdle, bar goes up.
STAMPEDE started in October 2005, and in 2011 we analysed the data emerging from STAMPEDE for the second time. At that time it was clear that arms D and F weren't effective enough to justify us continuing to enter patients into those 2 arms. So we continued to enter patients only into arms A, B, C and E.
This graph is one way of depicting recruitment to the trial. The x-axis here is year of recruitment and the y-axis depicts each of the arms in the trial. Now, each of these little dots is a man who's joined the trial. So you can see that we started very slowly deliberately slowly in only a few centres, and then we opened out to more centres, to the point where now we've got an increasing rate. Recruitment has increased exponentially, and we now actually have more than 7,000 men who've joined the trial. You can see how when recruitment stopped early to the celecoxib comparisons, the recruitment becomes slightly more dense to the other arms. So that was a benefit of stopping early.
And as you can see from this graphic, we've added 3 new arms to STAMPEDE. Recruitment to the original arms has stopped, while recruitment to the new arms, of course, has continued. The advantages of adding an arm to a multi-arm, multi-stage trial are principally twofold. The first is that rather than waiting to set up a new trial to evaluate your new treatment, which is demanding evaluation, you can just add it into a protocol that's already there and the trial that's already ongoing. So that's the first advantage. The second advantage is that recruitment to a trial often takes a deal of time to build up and get at full steam. By plugging this new arm into the ongoing trial, you're already at full steam, so you finish the trial much more quickly. So you get your answers much more quickly as well. So in the STAMPEDE trial, we'll have answered 8 questions in 15 years. If we had put those 8 questions as 2-arm trials, even if we'd done that in the most efficient manner possible, it would have taken us more than 40 years to answer those questions.
So to illustrate, with abiraterone, we started talking to Janssen about having access to the drug in 2010. We got agreement to go ahead in 2011. The French EORTC group got agreement to go ahead at the same time. So we both had got to the point where we could start the trial in 2011. We took 2 years to 2013 to recruit just under 2,000 men to our abiraterone comparison. It took the French group 2 years to get their first patient in because they had to set up the trial from scratch. Looking at it another way, it took us 5 years to open our first 80 centres. By the time we had the abiraterone trial good to go, we had 120 centres open. It took us under 6 months to get all 120 centres recruiting, as compared to 5 years to get 80 centres recruiting. So as a way of setting up a trial, it is just so much quicker. You get rapidity of setup, so investigators like it, trusts like it, because they don't have to stop and start new trials. And the trial funders like it because we get an answer much, much quicker than by setting up a new trial the conventional way.
No, there'll be none of my patients up there today.
So, multi-arm, multi-stage trials have a number of important advantages over the traditional 2-arm approach to randomised controlled trials. You can evaluate more treatments in a shorter time. You're more likely to find at least one treatment that is better than the current standard. They are lower cost when running multiple 2-arm trials, and they give you the flexibility to drop less promising treatments and add new ones that emerge. But when is it appropriate to use the multi-arm, multi-stage design?
We're using the multi-arm, multi-stage approach in a number of diseases now, and common to all those diseases is the fact that for many years we've made no real progress in treating patients with that disease. And there are many new treatments demanding evaluation. So we're using the multi-arm, multi-stage trial approach in diseases like advanced prostate cancer, in Alzheimer's. We're looking to see whether we can apply it to multiple sclerosis, and we've also applied it to tuberculosis.
From a researcher's perspective, there are strong arguments for doing more multi-arm, multi-stage trials. But what do patients think about that design?
The multi-arm, multi-stage design of trial is attractive to patients, I think, because they get the results early. I'm not entirely sure that if one was a patient, you actually are concerned about the structure of the trial. You want the results, and you obviously want them as quickly as you can get them. And a multi-arm, multi-stage trial does that.
So the key benefit of MAMS trials is they allow us to improve treatment for patients as quickly as possible. And if we look at trials like that, a MAMS trial becomes a no-brainer